from a very simplistic mindset, if you think about what is cancer, well, it's a proliferation of cells where these cells have acquired gene mutations. Every single cancer has gene mutations within these cells. So the source of the problem is the gene mutations. And if you could somehow find a way to just correct those gene mutations, well, that would fix the cancer. And it's easier said than done, but in theory, that is kind of should be the cure for cancer if we had that degree of ability, the ability to correct all these gene mutations. And so that kind of was, and probably still is, my very simplistic thought process on how to fix cancer. And it just so happened, I went to med school and went to residency, and years later, I was interviewing for fellowships, and I went to one of the fellowship programs, and the fellowship director was interviewing me, and he asked me what I thought at the time was a really hard question, and he said, if you were the director of the National Cancer Institute, and I gave you get $3 billion. That, for some reason, I remember the number he gave me. It was $3 billion to invest, but I said you could only invest it in one area of research, or one area to sort of cure cancer. What area would that be? And naturally, I had this sort of background, and I said gene therapy. And as I mentioned in the column, there had been bad press. There had been a patient with an inherited disease that had died during a gene therapy trial at the University of Pennsylvania. I think that had led the FDA to shut down the gene therapy research at University of Pennsylvania temporarily, and so it was kind of getting a lot of bad press. But here we are now, 25 years later, and we're starting to see the emergence of some real gene therapy, although it's sort of in a different form than what it was looking like 25 years ago. And so what I point out in the column is that there's some interesting science going on now with gene editing. Who could have foreseen the emergence of CRISPR-Cas9 technology and gene editing? But that's really what we're seeing now, is that technology is starting to make its way into the clinic, and certainly into clinical trials, and having an impact. And perhaps the first of the example of this is Exa-cel, which is not right now an oncology drug, but it was shown to have real benefit in patients with sickle cell disease and beta thalassemia. And what it is a cellular therapy in which the genes of the cells are modified by the CRISPR-Cas9 editing technology that edits the erythroid enhancer element of a gene called BCL11A. And what that does is it leads to disruption of BCL11A expression, which in turn leads to increased production of hemoglobin F. And we've known for a long time that patients with sickle cell and thalassemia who also have increased levels of hemoglobin F have much lower clinical effects, or clinical symptoms of their diseases. And so that was the rationale here. And sure enough, it really worked in these clinical trials. The patients with sickle cell disease had almost total elimination of their pain crises. Patients with thalassemia had elimination of their transfusion dependence. And so this product has now been FDA approved. And I think it's a little bit to be seen how it's going to roll out and be applied. I think the estimates are that relatively low numbers of sickle cell patients will have access to this treatment or will be candidates for this treatment. It's fairly intensive, but it's a proof of principle that gene editing now is going to play a role. It's not gene therapy by itself. It's sort of gene editing in combination with cellular therapy. But in oncology, we have another example that was published in the New England Journal, where the authors edited CAR T cells by removing certain proteins on the surface of these cells by editing the genes. And so I talk about that in the column and show that these CAR T cell therapy, this CAR T cell product was effective in a couple of patients with T-cell ALL. Not mentioned in the column because it was just reported this morning, but I just read an article that the protein CD5 may have some inhibitory effect on T cell function and may inhibit the efficacy of CAR T cells a little bit. So some authors just published in Science Immunology editing out of CD5 from CAR T cell products and in the lab in animal models that improved the efficacy of the CAR T cells by editing out CD5. So I mean, I'm sure we're just scratching the surface of the potential impacts of gene editing in cancer treatment. But you know, you can certainly imagine now with these newer technologies and ability to do this, to edit genes, that cellular therapy will be more effective, and that you know this type of technology will have an impact in development of new cancer treatments in the next decade or two. So I think we're right at the beginning of that, the emergence of this as new therapeutic modality. So I think it's really exciting, and maybe my answer, while it seemed stupid 25 years ago, you know, wasn't all that bad. So